as a basic immunologist and a translational immunologist, you highlighted very nicely this idea that immune systems are really dynamic events and multicellular events. The T cell is an important cell in the initiation and final outcome of that immune response. What are the other cell types that you think, as we move forward in cellular therapy, that we might want to also involve in that? And is there a role for things like synthetic biology in initiating these events?
Yes, I think synthetic biology helps us to understand the different components, how they interact, and allows us to take parts of that biology and synthesize it into something new. But the complexity is considerable. So we're looking at two main types of different cell type to assist T cell function. First is invariant chain NK T cells, which have much better ability to traffic within tumors than conventional T cells and recognize different types of molecule in association not with MHC molecules but with C1d and on monocytes and other antigen-presenting cells. Those look very powerful in their ability to change the shape of the immune response that occurs within a tumor, so that if you start attacking a tumor, it produces a whole lot of defense mechanisms like myeloid dendritic cells and tumor-associated macrophages that turn off the rest of the immune system. By using iNKT cells, you can interrupt that dynamic change and make sure that the tumor doesn't evade the attack by the primary T cell. And the second area that we're looking at is NK cells, which do seem to be resistant to being rejected by the host and which do not themselves cause graft versus host disease. So these may be very useful initiators of an immune response that you can then follow up with the T cell. So the complexity is high, but I think, as you said, if you adopt a systems biology approach and understand what bits are influencing which other bits, you can come and get the key components, the key opinion leaders, if you like, and feed them in at the right time.
That's really well put. So from a fundamental scientific standpoint, that makes enormous amounts of sense. At what point do we begin to get regulatory buy-in to try these kind of trials that can bring in other cell types and initiate system-wide responses?
So I think you've hit on a major question. I think the two difficulties are how do you regulate, how do you develop studies that use multiple cell types, some of which will never have been used in humans. How do you develop the studies that will tell you whether those are safe and effective? How do you pay for those expensive studies? And how do you get the companies to work together, each of whom owns a different piece of the action, and none of whom are too keen to devalue their piece in order that somebody else can use theirs. And those are difficult questions to which I think we don't yet have an answer. I think we can begin to see how the answers are going to evolve with the checkpoint antibodies used in combination with cell therapies and used in combination with small molecule drugs and so on. But that's taken a little while to emerge. I think what will push it is success. I think as people find things that work, combinations that work, there'll be an impetus, a drive to try other things and see if they work. But without that, it's going to be difficult, no question.
We're living now in a renaissance period where it's becoming mainstream with the acceptance of CAR T therapy as FDA-approved therapy and the myriad of really exciting clinical trials that are out there in this space. Could you comment a little bit on the technology that you've developed and you're focused on, which is the virus-specific T cell?
We started with virus-specific T cells because viruses were the main problem in patients who had bone marrow transplant. And the two types of virus they get are those that cause acute and severe viral infections. The second type that cause a kind of malignancy to develop, the EBV, lymphomas and lymphoproliferative disease. We started out really treating both of those kinds, but our primary focus was on the malignant cells that were produced by Epstein-Barr virus.
Some of the early work was pioneering in that Epstein-Barr virus specific field. As you take those forward in multiple clinical trials for other types of cancer, could you talk a little bit about the unique properties of your virus-specific T cells as they relate maybe to other conventional CAR T therapy that's been introduced in the past maybe 10 years or so?
Yes, well, CAR T cells, chimeric antigen receptor T cells, obviously have gained a great deal of publicity and attention for obvious reasons, very potent in hematologic malignancies. But I think we have to bear in mind that CAR T cells have had 25 years of intelligent design behind them, whereas native T cell receptors, the T cell receptors that are involved in treating virus infections and virus-associated cancers, they haven't had 25 years of intelligent design. They've had 250 million years of evolution and natural selection. And intelligent design can only go so far, however intelligent that design might be. So I think what we have with the native TCR is a number of properties that aren't possessed by these synthetic receptors. The most obvious is that they can recognize internal antigens within the cell, provided they're processed and presented on the cell surface. That's something that you can, in principle, do with CARs, but is very difficult. Really recognize quite as well is that they interact with the whole rest of the immune system, and they interact with it to broaden the reach of the immune system, to broaden the different things that it recognizes, and to grab in other cell types of the immune system that can produce different functionalities. And we realize that that's more and more that's important for the treatment of human solid tumors, because human solid tumors, unlike the hematologic malignancies that have been so successfully treated by CARs, human solid tumors are highly heterogeneous and they're highly plastic. So they're changing all the time and they're different at different times, and even in the same place. If you couple this with the ability to recruit many of the body's own defense mechanisms against the immune system that we use normally to protect ourselves from being destroyed by our own immunity. If you couple all those things together, you can see that a single T cell targeting a single antigen on a tumor, solid tumor, is not going to eradicate that tumor. It really takes a village. You have to have a lot of specificities and a lot of different cell types involved. And really, only conventional T cells can do that as easily as we would like.
With regard to the NK cells and other cell types as well, some of the advantages are in this allogeneic space. The ability to have an off-the-shelf product. This addresses problems in manufacturing, addresses problems in most importantly, addresses problems in therapy and treatment. Could you talk a little bit about the role of VSTs and your virus-specific T cells in this allogeneic space, and whether or not there's some promise there?
Yes, I think there is promise with virus-specific T cells. That the two problems with allogeneic cells, as you imply, are that they have their own T cell receptors which can cause graft versus host disease. They also express a great many different polymorphisms that can be targets for the host immune system to reject them. So you have this balance between the graft rejecting the host and the host rejecting the graft. You can take one element of that out of the equation with a virus-specific T cell, because the T cell receptors are, by definition, virus-specific. And it turns out that in humans, those virus-specific T cells don't have significant alloreactivity. And I think we've shown that over and over again in many hundreds of patients with many different viral targets. Rejection is a little more of an issue still. What seems to happen is that you have a period of engraftment followed by a period of rejection. But during that period of engraftment, because these T cells engage host antigen presenting cells and stimulate host virus-specific T cells around that same antigen presenting cell, you get, if you like, a second wave of recipient-derived T cells, so that they go in, they sacrifice themselves, if you like, the forlorn hope, as you might call them, and then they generate a recovery from the other T cells. That said, we are manufacturing now T cells which have got defense receptors on them that will attack and eliminate anything that attacks them first, enabling them to persist longer. I think it will improve, but already we can see significant benefits from third-party virus-specific T cells in really quite serious virus infections and tumors.
If you looked at the entire space, whether it's virus-specific T cells or CAR T therapy, oncolytic viruses, checkpoint inhibition, what do you see as the next promising combination, even therapy, that will be seeing some kind of effect in the clinic, whether it's cytokine biology or what is it, trying to initiate that system-wide response that you mentioned?
I think there's a lot of areas that could work. The problem we have, as everybody knows, is that once you start having combinations of things, you essentially have no good way of modeling them. So you can find a model that predicts something will work, you try it in humans and it doesn't. You try something in humans that works, you go back to a model and you find it didn't work in that model. I'll just give you one example. When we started our EBV T-cell trials to treat immunoblastic lymphoma in patients after transplant, there was no animal model. We went straight from the dish, so to speak, into the humans, and it worked beautifully, very well. About 12 years later, when someone finally developed an animal model, we tried it in the animal model, and all the animals died of severe graft-versus-host disease. If we'd had that animal model, we'd never have used it on the patients. So I think that remains a huge handicap to anyone who wants to try combinations. You can always come up with arguments for why they should be used and find studies in vitro and in vivo that suggest the positive outcomes, but the reality is that these have to be sequential, iterative studies. You come up with an idea, you try it out in a mouse model, if you have one, you try it in a patient, but it's really only in the patient that you can find the answer. I think things will get better because the depth of analysis that's now possible of events in vivo, in patients, gives so much more information that it gets much easier to predict back what's going to work and what changes you need.
Right.
But it's tough. That's a very hard question.
To your point, exactly. You have to study the system and really to understand the system itself.
Yes.
And the opportunity to do that, I think the clinical trials that you and your team have done really provide a huge opportunity for people to immune monitor, to really get an understanding of what the mechanism of action is when it does succeed,
Yes.
what the mechanism of resistance is. And then also to maybe look at other fields, like if you're studying allogeneic field, we could look at things like bone marrow transplantation and successful engraftment there to look at mechanism.
I think regulatory agencies are more and more aware of that problem, and they're very willing to allow these kinds of adaptive designs.
Thank you, Malcolm. This has been a great conversation, and I think, certainly speaking for myself, I've learned an enormous amount, and hope that the audience has as well. Thank you for taking the time.
Well, thank you. I always enjoy speaking with you, John.
Appreciate it.
Very impressed.